So welcome to our class on your unique MO. In other words, we're going to be looking at sensation and perception today. And you read a whole section, a part of your textbook about that in preparation for this class. Okay. Now, the first gateway to perception is attention, the gateway through which all sensory information must pass to be processed and experienced by you, right? And here's a really great review question. What is the second gateway through what most sensory information must pass before you actually experience it? It's a great review question because it is a brain structure, a brain structure. Can you think of it? And that would be the thalamus. The thalamus is, a great, you can think of it as a way station through which all of the stimuli from these different systems, sensory systems, must pass in order for it to then be interpreted and experienced by you, right? Interpreted by the higher brain and experienced by you. So attention is the first gateway, but attention is really selective, right? So usually selective attention is a great thing and it's quite functional and it helps us to focus. But sometimes it is not so great. It's amazing how much we can sometimes miss right in front of our faces. This is called the inattentional blindness. Inattentional blindness, okay? And so when we miss something, it's right in front of our faces. Now, imagine that you, like this guy, were talking to somebody at a service counter, and in the middle, they had to bend down for something, and come back up as a totally different person. Do you think you would notice that? You would think so, right? But this study found out that most people didn't. And what I'm going to show you, this video footage, is after they did this with this guy. They brought him back and showed him the videotape to get his reaction, all right? Saying, hey, you didn't notice that the person who was serving you switched right in the middle, okay? So here's him watching, they're showing him the tape itself.
So the next time you're positive about what you think you saw, take a moment to reconsider, because as we've shown you, what you think you see is not always what you get. Wow. All right.
So in this case, it was actually somebody with dark hair who was serving him. And when they came back up from under the counter, supposedly, it was somebody with blonde hair, right? Totally different person, all right? I want to dive right into a set of TYLs, test your learnings, to see how well you processed in this information in your reading in preparation for this class. So this is a set of two test your learning activities. And the first one is testing you on the stimuli of the sensory systems. This is a pretty quick one, so you get 1 minute to see how many you can get correct. Also, as always with these TYLs, you can always go ahead and get the easy ones first, right? Some of these are more easily intuited. And then work on the more challenging ones. You get 1 minute, see if you can match these up correctly and go. Okay, let's see how you did on this. Hey. So before we go over the answers, let's review something important. The somatosensory system is more inclusive. So it includes the skin, the muscles, and the joints, while the tactile system only involves the skin, the sense of touch, tactile. Okay. Another thing is you might have noted that there are 6 questions and 5 answers on the slide. Meaning that 2 of the systems share the same stimuli. All right, so here go your answers very, very quickly. 1 is touch. 2 is light waves. Number 3 is soluble chemicals, soluble chemicals. Number 4 is sound waves. Number 5 is head movements. Number 6 is soluble chemicals, meaning that the gustatory and the olfactory systems share the same stimuli, which are soluble chemicals. All right. So this is your second TYL of the set today. And this time it's covering receptors and sensation. So it's supposed to begin with the number 7. Keep in mind that when we say cilia, it's in a few of these answers here, that remember we're talking about tiny hair-like cells that generally line membranes in the body. So again, this is a quick one. So a 1-minute challenge and go. Okay, let's see how you did on these. Number 7, the answer is cilia in the cochlea. Number 8, rods and cones. Okay, number 9 is taste cells. And number 10 is cilia in the semicircular canal. Now, the semicircular canal is also involved in balance. So sometimes as people age, this canal dries up and that can really hurt their balance. Number 11 is cilia in the nasal cavity, and number 12, nerve cells on the skin. So now let's take a closer look at each of these sensory systems. The stimulus of the visual system is light waves, right? And when we talk about waves, like these light waves, they are measured in a few basic ways. So the wavelength, the wave height, which you both see pictured here really well, okay? And then purity is the third way. Now the wavelength is measured in lumens. So it's experienced as a brightness and it's measured in lumens. So if you're looking for the brightest reading light possible or the brightest light for makeup application, something like that, or for drawing, you would want to judge these lights in deciding which one to buy by the lumens, okay? Now, the wave height is the hue or the color. So here we've got the wave height, and this is experienced as the amplitude, or I'm sorry, the hue or the color, and it is measured in nanometers. All right. So now the purity of the wave, notice that I'm not going to give you one way to measure it because there are actually over 100 ways possible. But I do want you to understand what it is. And what it means is, in this case, how saturated is that light wave with light itself, how saturated. And what do I mean by the saturation of light waves? Let me tell you this. I could paint the same image once with watercolor and once with oil paints. And I could paint each using the same wavelength and the same wave heights. But that would still be, they would look very different, right? Which would be the more saturated with color? And that would be the oil paintings, right? Okay, so watercolor is watered down. There we go. That's what we mean by the purity. Now, here's something we really take for granted. How is it that we can see color? How is it that we can enjoy a fabulous sunset? Hey, or what about a beautiful face? What about a beautiful piece of art? What about this slide? So what is color vision? The theory of color mixing is called the trichromatic theory of color vision. And it involves three different kinds of cones, ones that respond to red, ones that respond to green, and those that respond to blue, respectively. Okay? The way that we see all other colors is accounted for by light wave mixing of these three. Sort of how an artist mixes paint colors on a palette, although light wave mixing produces different colors. You can see them here in the middle. So think about it, different amounts of just red, green, and blue accounts for all of the colors and shades of colors that we are capable as human beings of seeing. Actually, humans can only see a portion of the entire wavelength, only about 400 to 700 nanometers. Okay, many insects can see less than 200 nanometers, and many fish and reptiles, for instance, can see wavelengths that are much higher than 750. It's really interesting to think about humans' limitation in color vision. Just what other colors do many other creatures see? We can't even imagine colors that we have never seen. So there are ways in which some human beings are even further limited, and this is called color blindness. There are two different kinds, and they're spelled right at the bottom of this slide for you. Dichromats have two cone types missing. To remember, rods and cones are the stimuli of the visual system, and cones are responsible for your color vision. Okay, so dichromats have two types of cones missing. The ones that process red, the ones that process green. One of those are usually the one that's missing, right? So they tend to get their colors mixed up a lot. Okay, monochromats, who are missing two different types of the three cone types, they see words only in black and white and gray, different shades of gray. All right. Now, here's a great little color blindness test. See if you can identify the dog and the boat in this image here, in this matrix, within 5 seconds, 5 seconds, and go. Okay, so if you saw the boat by now, you are not colorblind. If you didn't, then you may be, and you may want to get this checked out. So I'm going to outline the image here of the boat with my cursor, and there you go. So now, if you found the dog in this matrix, that means you are very susceptible to the power of suggestion, because there is no dog in this matrix. All right. So how did the trichromatic color vision theory, how did it pan out? Actually, it panned out pretty well. It accounted for almost everything that scientists had discovered about vision today, except for one thing: after images. So an image and after image is an image that persists after extreme or prolonged exposure, okay, after the original stimulus is gone. And I'm sure you've all seen an after image before, but I want you to visualize a great demo that we typically do in the classroom. So we just did the color blindness test demo. We can do that on Zoom. And the rest of them that we do today, there are only going to be 2 that you need to visualize. The rest can be done also on Zoom, but this is one to visualize. So the first thing that we do is we train students to keep their bodies really still and to keep their vision really focused. And there are 2 ways that you can do this well. First is you could slouch in your chair, slouch down until the back of your chair functions as a headrest, like I did right then. Or you could use your desktop to rest your elbow and then rest your chin in your hand. And that will keep your head steady too. But I'm not going to actually do that because we're in the middle of a pandemic. And so I don't want to touch my face when it is not necessary. But know that either way will work, right? And so the first thing that students do is they get into their position so that they can keep their body really still, their head really still, and their vision focused. Okay. Then we turn off the lights completely, and students actually study this image for 2 minutes. And I'm putting this on the slide so you can do it yourself, but if you would like to tonight, but it has to be done after dark, when you could turn all the lights off in the room and simply look at this slide. Assume your position if you try this by yourself, and then set a timer for 2 minutes. Look at it for 2 minutes, focusing on that center dot right there. See where my cursor is between the eyes. Okay. And then when we switch to this next slide, keeping your vision right on the slide, but now focused on this dot in the middle, what students see is the after image form around it. Okay. And there are a lot of exclamations through the auditorium of students as their after image kind of fills in, because the after image is actually a full color photo of Beyonce. And it's beautiful. So you can even do this at home. Again, if you want to use this prior screen and this screen, just advance to this screen at the end of your 1 minute timer. And you know that you can slide through the video once it's posted, our class recording. You can use a slider to find these 2 particular slides right away. Super easy. All right. So you read about the additional theory of color vision to explain after images, and it is called the opponent process theory. And it goes like this. After images are accounted for by the 3 different channels of cells in the occipital lobe. Okay, each channel processes a color, as you see on the slide, in light waves, and another color that is the opposite color in light waves. Okay, so one channel processes black and white, one processes red and green, and the other one yellow and blue. Okay, now prolonged or extreme exposure fatigues the cells that are firing. So if you're looking at black, right, it's going to fatigue those cells, and they're going to start firing inhibitory. Okay, remember changing voltage to negative inhibitory, which stops them from firing. So these are getting fatigued, getting tired, and as they are inhibited, fewer and fewer of them are processing because you're becoming habituated to the image. You don't need to keep having all of them firing, right? So some of them are inhibited. And what happens when we take the image away is that the ones that are inhibiting, they're making the other cells remember, lower than a negative 70 millivolts, right? They, well, a good way to think of it is that there's a lag time before they stop firing those inhibitors. So in that lag time, what you see is the opposite. So if you were looking at black, you see white. If you're looking at red, you see green. If you were looking at yellow, you see blue. All right? Now notice an extreme enough light wave can also do this, even if it's not prolonged. So if you turn around and look directly at the white projector light in a movie theater and then look away at a blank wall, for instance, you will see that that dark spot, that black spot it makes in your vision. So you've looked at this one, right? And you can see the after image in black. All right, now this also means that if you remember these 3 channels, you can predict that this green, yellow, and black flag pictured will produce a vivid red, white, and blue one as its after image, which indeed it does. And here's that slide for you if you want to try it tonight on your own. So which theory wins out, right? Well, both of them do. The theory of color vision accounts for how we perceive color. Remember, that's color mixing theory, right? Trichromatic theory. And the opponent process theory accounts for how we perceive after images of extreme or prolonged stimuli. So now don't forget to print 2 copies of the figures that you got with this part of reading that you did in preparation for the class today. So you were given a couple of figures and you want to print 2 copies, remember, so that you can black out these labels on one of them and then staple them back to back and use them as a study sheet. So you can test yourself, flip them over and see the answers. Really efficient, quick way to learn these, all right? Now, when we perceive the world, we are experiencing it through our own unique lens. And those lenses are affected by so many things. Yes, they're affected by the light waves coming in. They're also affected by past experiences, by your biology, your chemistry, expectations you may have even. It's easy to see that we each have our own unique MO for perceiving the world, with some things like ink blots and images in clouds that are clearly ambiguous, right? But not all ambiguous stimuli appear so clearly to be ambiguous. So, like for instance, this famous, a white and gold wedding dress. A young woman posted this dress that she purchased on the wedding, for the wedding on Facebook. And remember, if you've heard about this, her surprise when she found out that half of the viewers saw it as she did, it was white and gold, but the other half saw it as blue and black. Well, which is it, right? We're looking at the same slide here, okay? We're all looking at the same slide. And the same light waves are playing on our retinas and being interpreted back here, right? So why is it that 50% of the class in the classroom will raise their hand that they see a gold and white dress, okay? And the other half will raise their hand and say they see a blue and black one, okay? Now, let me actually show you what everybody's seeing here. All right, so the leading theory in this particular case is that when the picture is not the best quality, which this one is not, the brain assumes, the one that I showed you on this previous slide, it assumes the ambient lighting. It is assumed to have been photographed in the daylight, okay? When the picture, if your brain assumes that it was actually seen in bright daylight, it is seen as the blue and black dress shown here, okay? And if it is perceived to have been like in ambient shadows at nighttime, evening shadows, you might say, then it is seen as the white and gold one over here. And that is why, looking at the same slide, about 50% of you see this with the prior slide, something closer to this, and the others about something closer to this, okay? My friends, remember that perception is highly subjective and complex. So there are so many other things that also can influence which color we saw on that prior slide. So this is just one example. When it comes to color receptors or cones, we have the lowest percentage of blue. So remember, we had 3 types, right? And one type was the ones that process blue. We have, as humans, the lowest percentage of them. Also, we vary individually as to how many of those we have. So if we have less than normal, it nudges our likely perception towards this white and gold on the left. If we have more than normal, it nudges it toward the blue and black on the right. There is an equally famous ambiguous auditory phenomenon too. And what are the 2 words? If you've heard of this, then you might remember that they involve the word Yanny, right? Yanny and Laurel. Now, some people hear Yanny and some people hear Laurel when listening to the exact same recording. So we have a series of virtual labs toward the end of this class, and one of them explores both of these ambiguous phenomena in even more detail. Now, I want you to visualize the last demo today that you need to visualize. So only 2 of them out of the many we will do today. Do you need to visualize? And so they can't be done on Zoom. And this one might be the freakiest demo of all of them. Okay, it's an optical illusion and it's really cool. So visualize this as though we're doing it in class, because you will have an exam question referring to it that way. But first, students assume their position again, whether it's slouched back with their head resting on the back of their chair or whether they're putting their chin in their hands. And we turn off the lights and students stare at focusing in the very center. They stare at this moving image for only 1 minute. So again, if it's pure dark, it has to be really dark to do this. You can do it at nighttime. And here's the slide that you would use for that if you wanted to do that. All right. So as we turn the lights back on, when the timer goes off, students are instructed to immediately look at the back of their hand. Okay, look at the skin on the back of their hand. And it causes the illusion that your skin is crawling. And if you were to look really, really closely before it fades away, you would note that it is actually crawling in the opposite direction of the video. So you have neurons in the original occipital lobe that respond to movement patterns too. When you first start looking at the spiral, they are firing like crazy, but then they gradually slow down firing by inhibitors, some of them being inhibited, right? As you habituate to this, you don't need all of them firing all the time. Your brain can kind of fill that in. And so some of them are getting tired, some of them are getting fatigued, and they are stopping, right? Because of the lack of change. So adaptation occurs, and by 1 minute of exposure, most of these have generally quit firing altogether. When you stop looking at the spiral, turn the lights on, look at the back of your hand immediately, the pattern detectors are still not firing. The inhibitors are taking over now, right? Because the original stimulus is not there any longer. The inhibitors are taking over. And what you are seeing is the opposite movement. That is the illusion, creates the illusion that your skin is crawling, actually crawling on your hand. Right? So when you stop looking at this, right, this is what happens. If the original image, like this one, is clockwise, then the after image appears to be counterclockwise. Okay, so let's do one more demo before we move on from the visual system. We can do this one right here on Zoom. So here's the deal, guys. When I change a slide, you are going to want to count the F's in the sentence as fast as you can. So you're going to be given 5 seconds to count them, and then we will poll to see how many you found. Ready? And here we go. Okay, now I'm going to poll for this, and I want you to put your answer in here. Okay. Move your answer into here as quickly as you can. Okay. Looks like most people have their answers in now. So what I'm going to do is to go ahead and share the results with you who are with us here on Zoom. Live will see this. If you're not here with us in real time, you watch the recording, you won't, but I'll talk about it. So here are the results, okay? And you'll see that, as we typically get, 3 or 4 is usually the typical answer. And in this class, the most people voted for 3, saw 3 of them. But we also have a number of people who saw 4, some who saw 5, some who saw 6. What's up with that, right? What is up with that? Well, let me tell you what this means. Oh, here we go. If you saw 3 of them, you are normal. You did normal on this task. Okay. If you saw 4 of them, you did above average on this task. And if you saw 5, you are well above average. And if you saw all 6 of them, okay, then you did amazing on this task. So yes, there were actually 6 F's in that sentence. Don't believe me? Let's look at it again. Some students still cannot find 2 or 3 of the F's until we actually draw their attention to the fact that there are 3 instances of the word of, and then usually there's a big oh out there, right? So some of you probably are now going, oh. There's a fascinating explanation for this. The brain learns to skip small words to increase our reading speed and efficiency once we are fluent in a language. It becomes familiar enough with the context of particular small words. It skips them, and the brain fills in the missing information on its own. And that is also why those of you who are not fluent with the language yet actually tend to do better on this task than those who are not. Okay, now you read about some Gestalt principles of perception in your online reading this week. This demonstrated one of these principles in perception. Which one? The answer is, I'll give you a hint first, starts with a C, okay? The answer is closure. So it's a great demo of closure. Hey, the brain, and that is defined as the brain tends to fill in missing information that gives closure or completeness, right? All right. So moving along here to the auditory system. Now, the auditory system stimulus is sound waves, and they are also measured by wave height, wavelength, and purity because they are waves also. They're just sound waves now, right? So the wave height is perceived as amplitude or loudness, right? And that is measured in decibels. The wavelength, okay, is perceived as the pitch, and it is measured in hertz, Hz. And the purity, remember, can be measured in very many different ways. But the purity in sound waves is the timbre. It's called the timbre, and it still just means how saturated is that wave. Okay, but now we're talking about how saturated in sound. So imagine that I play a middle C on the piano, and then I play a middle C on the flute. Even though I play them both at the same wavelength or pitch, being the middle C, and I play them all at the same wave height or amplitude, they would still sound very different, right? That difference is the timbre or the purity. So the flute sounds less pure than the piano sound because it is more breathy, right? Okay, so human hearing capacity is approximately 20 to 20,000 hertz. 20 to 20,000 hertz. Now, some species like bats and porpoises can hear frequencies well above 20,000 hertz. And others, like homing pigeons, are sensitive to frequencies that are well under 10 hertz even, right? All right, now don't forget to print and study this figure of the ear, also provided with your online readings this past week in preparation for this class. And the ear, remember, comes in 3 sections. The outer ear, the middle ear is kind of more here, right? And then the inner ear here, all right, inside the brain. So the outer ear is composed of the pinna, see my hand here, the pinna and the auditory canal. Now the pinna is the somewhat cone-shaped fleshy structure on each side of your head. It is cone-shaped to catch the sound of waves and funnel them into the auditory canal. So if you look here, they're going through the pinna, is focusing them in through this auditory canal. Hey, and it is cone-shaped for that reason. Now, how many decibels result in physical pain for human beings? That would be about 120, all right? Okay, now between outer and the middle ear, we have the eardrum. See this little pink membrane here? Okay, that is the eardrum. And it results in hearing loss and tinnitus if it ruptures. And it may be temporary, but with enough of this happening or long enough, right, it could be permanent hearing loss. So how many instantaneous, how many decibels instantaneously rupture the eardrum? You read this and you might remember it's about 160, right? Now, another common cause for eardrums rupturing would be a combination of air travel and upper respiratory infections together, right? This combination prevents equilibrium of the middle ear. See where my cursor right now? The middle ear. Prevents the equilibrium of the middle ear pressure. Now, yet another common cause is Q-tips, and these are not meant to be cleaning out your ears like some of you may have heard and use them for. And they can actually really result in harm to the eardrum and the ossicles. There's actually a warning on the paper. What do I mean by ossicles? I'm talking about these tiny, cutest, tiny miniature bones in your body, okay? 3 tiniest, cutest bones in your body, collectively called the ossicles, and individually aptly named by their shapes. So hammer, anvil, stirrup. Okay. Finally, the inner ear consists of the semicircular canals and the cochleas. Okay. Now, as we mentioned before, the semicircular canal is involved in balance. Okay, the cochlea is the snail-shaped structure or underneath it. Okay, see this? And it is filled with fluid. The sound system receptors are the cilia lining the inside membrane of the cochlea. Okay. Now, the olfactory system, which is your sense of smell, and the gustatory system, which is your sense of taste, are collectively referred to as the chemical senses, okay? Because they share the same stimuli, okay, being chemical molecules. So remember that they share the same stimuli. And because they share the same ones, their interaction is particularly strong. So believe it or not, if you were blindfolded, if you blindfolded yourself and clipped your nostrils shut with a clothespin, say, and had somebody feed you pieces of apple, firm pear, raw potato, and raw onion, you would not be able to tell the difference. So most people cannot. Okay. Now, there is a freaky looking condition of the tongue that is called black hairy tongue condition. And in this case, normal shutting of the papillae does not happen, okay? And instead, the papi elongate and they trap bacteria. So your papillae on your tongue are supposed to, right, normally shed quite frequently. And this can actually be caused by many different things. So can be caused by smoking, can be caused by poor hygiene, but it also can be caused by some medications or a condition called dry mouth, when your mouth does not produce enough saliva. And it can be caused by sleeping with your mouth open. Okay? And the condition is freaky, but it's relatively harmless. Okay? If this ever happens to you, hey, you'll want to brush your teeth and your tongue frequently. So more frequently than you normally do, and it usually will go away on its own within a few days. Now, the tannins in some red wines can also turn your tongue dark purple or even black. So if your tongue shows a wine stain, brush gently, okay, extra gentle, because the acidity in wine can otherwise harm the enamel of your teeth. Okay, so if this ever happens to you from wine, you want to brush both your tongue and your teeth, but very gently. All right. Now, the tactile system is the sense of touch, and the receptors, remember, are nerve cells in the skin. The sense of human touch is so very important. It can be the deciding factor in whether or not a premature baby lives. And that's why incubators now have holes in them. And you can touch your baby if it's being held in one in the hospital. And you can even stay overnight sleeping in a chair next to your baby with your hand on your baby all night. Now, some studies have found that having a loved one hold your hand while you are experiencing pain can really help to buffer pain sensations. So it's real physical buffing that can happen. Okay. Phantom limb is another really interesting condition. It's the sensation of tactile coming from a limb that has been amputated, right? It's like this limb is not really there, right? It's been amputated at about here. But this person feels like they still have the lower part of their leg, the calf, and the foot, because they're getting sensations from it. Now, researchers have found that phantom pain involves actual activation of the nerves in the afferent pain fibers that are going to the brain from the amputated limb. In this case, remember, they're entering the spinal cord through the very bottom and then going on up to the brain in those afferent fibers, right? So the sensations that are experienced are from real activation. Okay? The phantom pain and phantom limb usually lessens over time. So can really drive you crazy, can drive people crazy until then, but you can at least let them know that it will go away over time. It will not last forever. A rare monogenic double recessive genetic disorder is called, for short, CIP. And that stands for congenital insensitivity to pain. And congenital means from birth. So no sensitivity to pain from birth. Okay, this involves dysfunctional pain pathways, but note that all the other sensory motor pathways, tactile pathways are intact. But people with CIP do not feel pain. Now, people with CIP tend to be very careless because of this. They have accidents. They usually die young. The adolescent boy pictured on this slide worked as a street performer in Pakistan. He would push knives through his tongue and arms on the streets, and he would actually walk on hot coals. A couple years after this picture was taken, he tried to impress his friends by jumping off a house. He felt no pain, but he died of internal injuries. Hey, now, so in this picture, you might think that, might at first thought, well, this is an illusion. He's not really pushing it through his tongue, right? Yes, he was really pushing that through his tongue. And especially when he removes it, there's a lot of blood, right? When CIP babies start to teeth, they sometimes bite off their tongues so they can also bite through the jawbone, right? So sometimes they need to have all their teeth removed because of this. Toddlers with CIP can blind themselves with their own fingers. Okay? Many other accidents cause loss of use of limbs and or death. So people with CID rarely live past the age of 25. Okay. What if the senses got mixed up and we experience numbers as colors, or sounds as tastes, or letters as colors? Okay, this happens with some people, and it's called synesthesia, which is defined as experiencing sensations in one system when another is stimulated. And it is due to some neural cross-wiring, either within senses or across senses. And the most common kind of synesthesia is experiencing numbers as colors. Now, here's a great test for synesthesia. So when I switch the slide here, I want you to count the twos in the picture as quickly as you can. Ready? And go. So most people have counted them by now and know that there are 6 twos in this picture. Now let's try this once more. Okay. So this is the same amount of time, but most people are still counting right now. Okay, because it takes longer to find them in this matrix where all of the numbers are in a black font. That is, it takes longer to count them if you do not experience this kind of synesthesia. Because if you do, you actually see the numbers, twos, in a different color font than the number 5s on this slide. Okay. So we can test for synesthesia really easily, right? With tests like this one, even when we change where the twos are, where the 6 twos are, people with synesthesia take the same amount of time counting the twos that quickly, right, as you did in that prior slide. All right. So interestingly, recent research has found that the colors associated with the letters usually coincide with the Fisher-Price magnetic letter sets, which many of us were exposed to at home or school during childhood, right? Okay, so far we've been looking at supraliminal perception. Supraliminal means that it is presented above the threshold needed for conscious perception, above that threshold. So you consciously perceive something. Subliminal means just below the threshold needed for conscious perception, but just below, so close enough to be perceived unconsciously. Now, many studies have attempted to determine if we can be influenced subliminally. For instance, the first study flashed buy popcorn messages on the screens at some theaters and not at others, and flashed them, hey, very, very quickly, thinking that too quickly for people to see, but so close to quick enough for them to that most people would subconsciously see it and buy more popcorn. And they did buy more popcorn than the control group, who did not get these messages at all. But there were many problems with this study, including the fact that it used the wrong control group. When replicated with supraliminal words in the control group, so instead of no message, they got a normal ad that stayed up on the screen, buy popcorn, right? Both groups bought the same amount of popcorn. In other words, hey, you may be convinced by a subliminal ad to buy popcorn, hey, with subliminal advertising, but only if you would be influenced by a supraliminal, normal ad, okay? And not if you wouldn't from a normal ad. For that reason, most companies just generally, they don't try this anymore because one, it's really difficult to do that subliminal window of how long it needs to be flashed is different for different people. And two, they know now from this research that only people that would be convinced by a normal ad would be convinced by the subliminal one. Okay. However, there are many graphic artists that are working for these companies that are found to be playing sometimes with these images without the company knowing it. Let me show you a couple of examples of that. And generally, the companies just take these down once they figure it out, right? That their graphic artists were just being facetious and someone actually points this out to them. But here's an example. Let's see if you can find the subliminal message. I think it's particularly easy now that we've talked about them, but people, if you just show this picture to people, they usually don't see it right away. In case you haven't seen it, I'm outlining the word sex in these ice cubes right here, right now with my cursor. Okay? And ice cubes, by the way, are a common medium for attempts at subliminal messages because they are super complicated images to paint or draw. And they can't be real ones, cannot be used oftentimes in these ads because the lighting needs to be too bright and they would be melting too much, right? Now, here's another example. And this can design was removed by Pepsi in the 1990s after it was pointed out to them that all of these lines are not exactly random, right? See the word sex? Hey, if not, watch my cursor. Whoops, I did not mean to do that. Here we go. S, E, and here's the trickiest one, X. All right. All right. So this concludes our lecture on our unique MO, how we perceive the world. Next week, we're going to explore sleep and dreaming. Sleep and the party that occurs when the boss is gone, dreaming.